Thank you. Good evening. I'm George Yazigi. I'm head of Dartec Digital Systems. Dartec is a research center at Cranfield University. It is focused on airlines, business, and aviation. And the word business, it means research, not really business in the strict sense. So one of the areas that I work on is artificial intelligence, machine learning, and high-performance computing. This is the main bulk. And also, I have lots of backgrounds in so many different things, so I'm not going to mention all of that at the moment. But in general, I also have background in genomics, and hence today I'm presenting to you this presentation, and hopefully you'll like it. As a word of warning, the presentation, I tried as much as possible to avoid technical jargons. So I'm not going to delve into equations. I'm not going to delve into any mathematical modeling or formulations. I'm just going to pass through smoothly around certain concepts. There are two main concepts that I'm going to try to stress on. Why bioinformatics and computational biology are mainstream now, and what really the advancement in computing has done to these two disciplines. And we will try to learn whether bioinformatics is achieving its promises, and whether we have something in the future, and what are the disadvantages. And mainly I'm going to be stressing a little bit about the ethical side of bioinformatics. So this is not moving for some reason. Let me see.
You may need to move the mouse to the presentation screen.
It's not moving for some reason.
Yep. The mouse should be on this side for it to move.
Oh, okay. So I can still use these. Okay, so as a disclaimer, please don't think that this is a course or any kind of material for bioinformatics or computation biology, because it is not. Otherwise, you will get bored immediately after probably 5 minutes. So the idea is to focus on certain main points. So what is bioinformatics and computational biology? Many people would confuse the two. Many people would say bioinformatics equals computational biology. This evening, we're going to really delve into that and see that these are two different, I would say, research areas which translate into two different streams of, I would say, disciplines. So bioinformatics is mainly focused on the analytics side of things. So imagine in every environment, when we have analytics, it means I have a large amount of data and I want to derive some relationships. So bioinformatics is the art and science of deriving relationships from the data related to biology, related to computational biology, and we're going to see how these areas are going to I would say cross. So computational biology is only focused on the concept of modeling. And basically, rightly so, Alan Turing in the 50s was calling this modeling, purely modeling. But now we call it computational biology because we have a lot of other, I would say, algorithms which have been mainstream in the market in order to help and alleviate the long times of waiting for those researchers who were trying to derive some information from the knowledge at hand. So when we talk about modeling, It means that we could also train some models using artificial intelligence, basically deep learning, and then we could use those models to map against reality. So, in other words, these are the two main pioneers of bioinformatics and computational biology. This lady, Margaret Dayhoff, she patiently worked about 11 years to just decode one sequence or one protein. She was interested in protein decoding or sequencing. She spent a lot of time, but she is the first person also to use computer languages to do that. And we're going to talk about that later. So Mr. Waterman was the first person to publish a paper, and he actually created the Smith-Waterman algorithm, which would help in the decoding and the sequencing of the DNA. So the word sequencing is a fancy word to say studying the order of things. So if I have A, B, then X, then C, this is I'm studying the order, why I have X instead of something else, okay? So the other thing is, we have also this guy, he was tremendous contributions, he did tremendous contributions in the field of computer science, and he was also based in Bletchley, not very far from here, and he also started working on the understanding of the morphology of proteins. Many people probably do not know that he was not only a computer science, but also he was working on biology. So he basically introduced the biological morphogenesis concept. In the 50s, computational biology was not there as a term. So he actually started that concept, but he did not name it computational biology, rather he named it modeling. And we have this lady, Pauline Howe, or however you pronounce the name, she's from the Netherlands. She is the first person to coin the word bioinformatics. So with her research partner, Ben Hesper, they together created the understanding of bioinformatics as the study of informatic processes in biotic systems. So the moment we talk about the study of a process, that means we have data and we have to analyze that data. That's the concept of studying something in this context. So forgive me about this, but I have to share it with you. Because if I don't share it, it's not going to be about bioinformatics and it's not going to be about computational biology. So what is that? In every person in our body, we have 20 amino acids. Amino acids is a fancy word for molecules that are basic and that are produced by the flow or by the process of creating creating proteins. So the concept is that each amino acid, and this is a molecule, just as we all know that we have H2O for water, it's a molecule of water. We have these 20 different amino acids. And these 20 different amino acids, these are the basic amino acids. They are basically divided or grouped into 4 main groups. I'm not going to delve into that, but just for your info, we have hydrophobic amino, polar amino with positive charges, and polar amino with negative charges. I'm not even going to delve into this because you're going to hate me for that if I go through those. But just suffice to say that in order for me to develop a protein, I cannot develop that protein unless I use amino acids. This is how the body works. And to be honest, every single molecule in our body is based on that concept. We do not, you imagine that these are like tools or basic building blocks with tools in order to build up the human body. So I cannot have a cell, I cannot have an organ, I cannot have a digestive system, respiratory system, you name it, nervous system. I cannot have these without amino acids and proteins. Now, and by the way, those of you who are a little bit interested, why I'm talking about this guy, cysteine, many people would misunderstand that, okay, I have a problem, I need to take some medication. However, sometimes if we take folic acid, it combines with other amino acids in order to create more beneficial environment in the bloodstream. So those of you who have not heard about a protein called homocysteine, the homocysteine could be increased by just taking folic acid. Why I'm talking about that, I'm going to mention that later, so please keep your mind on this. And here we call the 4 types of proteins. And you will know later why I'm mentioning this. These are the 4 basic, and there are no other. We have only 4 types of proteins. We have the primary, that means it looks like a ribbon, just a ribbon, no folding. We have the secondary, we start having loops in that ribbon. And then at tertiary, we have more looping and interconnections. And we have quaternary. It is the most complex, I would say, representation of a protein. Why these are important? I don't know why these proteins are affected by their forms. And this is what Alan Turing was trying to understand, the morphology of proteins. Because rest assured, if I have a different angle, you see this fold like that? If I have a different angle, then I have a different functionality. It is extremely sensitive. And you can ask me why you are mentioning all of this. This is not a biology class. Exactly, this is not a biology class. This is about computational biology. This is about modeling. Because if I understand all the structure of those proteins, I will be able to treat diseases. If I have a change in this protein, that means the patient or the subject is probably having cancer. And it is very important for us to understand this relationship. The form of a protein defines its functionality. And when there is deformation in the protein, there is deformation in the functionality, and therefore we end up by having problems. So every job in the body, every job is controlled by those proteins and is controlled by the structure, most importantly, the structure of the protein, whether it is primary, secondary, tertiary, and quaternary. So in reality, this is a proteoform. The proteoform is basically a unique form of a protein. I can have 2 glasses of water. They are similar. Here we have many glasses of water. But if I have another glass of water, but it has a different form. So morphology is very important with proteins. So a proteoform is a unique form that our genome, in other words, our DNA can produce. There are basically roughly 20,000 of those proteins in our body, produced by our body. But also, Some studies would say 500,000. I tend to say probably 400,000 as a maximum, but there are studies which say 500,000. So theoretically, the number of unique proteins that could be produced is infinite. And I'm going to show you that. So as you can see, from a very simple protein, I can have a more complex protein. I can have an organelle. Organelle is a part of the cell. I can have the cell itself. I can have a tissue. I can have an organ and a system. And from the system, I can have also a conglomeration of systems. So this is a very, very interesting photo that can show us the synthesis of proteins, proteosynthesis. Going from a simple DNA structure, here is the DNA, and then here where we have a disconnection by using the RNA will help to loosen the DNA structure. Then we can grow into an amino acid, and from an amino acid we can have the protein. Please leave this picture in your mind. Try to memorize it, because this is very important, because the angles between those folds is very important, because this will tell you whether this is a healthy protein or not. And if we do not have a healthy protein, we do not have a healthy person. So this is very essential to be understood. So the cycle is very simple in words, but in reality it is more complex. So we have a DNA, it's the double helix structure. The DNA is decomposed by the RNA and the mRNA, the messenger RNA. You must have heard that a lot when we had COVID, when they created the vaccine using mRNA technology. So the messenger RNA is helping in the production of the protein. And this is why the technology of the COVID vaccine, which was designed by Pfizer, was basically replicating the machinery of the virus by using our RNAs. So here we create the protein, and that protein is the building block of everything. Now, I hope you are not bored yet. So the process is still simple. It's the same process for proteosynthesis. We move from DNA to RNA to amino acid to protein. And the body is responsible for that. Actually, our DNA is responsible for that. This is why there are two reasons for problems in life in terms of psychosomatic problems. That means body-related problems. Either it is a DNA problem or a protein problem. I'm not trying here to oversimplify, but in reality, it boils down to these two, either DNA or protein. And this is where we can see, and forgive me for this chart, here we can see how the sequencing happens, the transcription happens, the translation happens, and then a protein is produced. Please don't worry about that. Just it is for you to realize how complex are these structures happening without us even thinking about it. It happens every second in our life. So at every stage, if something fails in the machinery, let's say if you are maintaining a car and you forget a spanner in the engine, what would happen? Okay? Sometimes these things happen also in cells. And this is where we can have diseases. This is where we start having problems. And of course, aging is also related. I'm not going to focus on these because really they are boring. So, how many proteins can be manufactured? Right. As I have mentioned, we have transcription and translation. That's the basic process, and there is no other process. If the machinery cannot transcribe and translate, so transcribe, it means to unloose or to unfold the DNA structure into two separate. Imagine you have a zip. When you unfold it, this is when you are transcribing. I'm saying that in very, I would say, simple terms, but the process is very much more complex. So transcription and then reverse transcription. And you remember people who, when the AIDS pandemic happened, it was related to the reverse transcription affecting the DNA of a person. So here I have the RNA. This is when the transcription happened. Then I do translate. What translation means? Converting the RNA into an amino acid. And the amino acid will become later on, any agglomeration of amino acids is basically a protein. So when I'm talking about how many amino acids we had 20. If I have, on average, a protein is made up of 400 amino acids. So the simple formula to obtain the maximum number of proteins, possible proteins, is 20 to the power 400. If you have a hand calculator, I think your hand calculator will give you infinity. It will block. It will not compute that number for you. So this is a very large number. This is 2.58 times 10 to the power 520. This is the number of different proteins that I can produce just by having 20 amino acids. But we have seen in the past, and even today, we have also certain proteins which are manufactured or produced in the body based on 1,000 amino acids. So the formula will give me the summation of anything between 1 to 1,000, but of course maintaining 20 to the power n, or 20 to the power of 1,000. So the number is huge, 10 to the power 1,301. That's approximately the number of different proteins. Why I'm mentioning this, it's not to tell you that I'm good at math, no. It's just to tell you that we need humongous computation power if we wanted to deal with this. And this is why people in the past, for example, Mr. Waterman, you have seen that guy, who is basically the father of the transcription. Mr. Waterman, he spent at least 10 years so that he could transcribe or sequence the genome back then. And the genome back then was not what we know now. So this takes a long time. This takes humongous efforts. We need to use advanced algorithms in order to make approximations, and I'm gonna touch on that a little bit. So what we end up with is our body functions, our traits, our strength, our well-being, basically everything in our body, I'm talking psychosomatic, is depending on two things, DNA and proteins. Why these are very important? Because if we can predict all the shapes of all proteins, we can potentially bring solutions to literally all diseases. And this is the challenge, whether we can predict those. And I'm going to touch on that later on. So this is how the number of possible proteins can be huge. This is why we need computational biology to create models. And this is why we need huge databases in order to have big data analytics, in order to analyze the environment and the context of interactions. So here, as you can see, this is a DNA. It has been transcripted. It becomes RNA, and then from RNA, we have the creation of the amino acids through the messenger RNA. It's like someone who's telling you, look, this is the code. Please do this recipe for me. This is the messenger RNA. This is the role of the messenger RNA. And then here I create the amino acids, and then I can have the proteins. And all of this happens inside our bodies at any time without us noticing. So, now back to reality. So what is bioinformatics? It's basically the intersection of biology, statistics, and computer science. When I have the intersection between biology and computer science, I have computational biology. The intersection of biology and statistics, I have biostatistics. Intersection of statistics and computer science, I end up with data science. So at the heart, the intersection point of all of these three disciplines is bioinformatics. So it's wrong to confuse computational biology with bioinformatics. It's very wrong. However, many people would do that. So, this is a little bit more complex. So this is the bioinformatics ecosystem. Those of you, if you are in a university, those of you who have studied physics, you could relate physics to chemistry and statistics. You could relate statistics with mathematics. You could relate engineering with computer science and with mathematics. But at the heart, excuse me, we have biology. And biology is intersecting with computer science to create computational biology, with engineering to create bioengineering, with mathematics to create mathematical biology. Maybe these terms are not taught, or these subjects are not taught here in the UK sometimes. I don't know. I know some of them are being taught. If we amalgamate biology and physics, we have biophysics, and with chemistry we have biochemistry, and so on and so forth. So all of this is bioinformatics. So the difference between the two slides, we have bioinformatics as the intersecting point between these three sciences or three disciplines. However, and computational biology is only the intersection between biology and computer science. However, in reality, the big picture is that bioinformatics is all of this ecosystem. Because there is a lot of data that needs to be analyzed. And each data coming from a data source or from a discipline, and they all contribute to bioinformatics. I hope now that I have clarified what bioinformatics is and what computational biology is. So, this lady, she was still young. Have you ever seen these punch cards? Have you worked on punch cards in the past? Anybody? Yeah, fine? You could sell? Yeah? So she wrote yeah, brilliant. So she wrote a Fortran program in 1962, probably. Actually, she wrote it in the 50s, because only in 1965 she was able to produce what is called her atlas of proteins. But she spent 11 years using comprotein. Comprotein is the program using Fortran language. Unfortunately, this language has no longer been taught at universities or even anywhere. However, we still use it, and it is essential in machine learning and deep learning. When you install, those of you who are familiar with machine and deep learning, when we install CUDA, which is the platform for using GPUs, which is the accelerators for computations, CUDA is essentially based on Fortran. but nobody talks about it. So she wrote that program in order to sequence one protein only. It took her about 11 years. But once she sequenced one, she was able to sequence the others relatively fast. However, in those days, it was very limited. It was punch cards, and it is a very tedious task to use punch cards. So, and why this matters, we will try to answer the question in a moment. Also, it took 13 years to sequence only 92% of the human genome. Probably a few years back, many people were bragging, we have deciphered the human genome. That's not true. They have only deciphered the first probably 12 to 15% of the human genome. And this 92% is the 92 perceivable. Because if you happen to study genomics, you will soon realize that science would say, or would claim, that the genome, 98% of it is nonsense. It's only 2%. But this is not true. Nonsense because it's not discovered yet. So the 92% is what actually discovered. And Waterman was working on a sequence algorithm with another guy called Smith, and this is why we have the Smith-Waterman algorithm. This, instead of basically running a long sequence, a long discovery, imagine if you are scanning. Let's say you have a torch, and you want to move that torch along every single point in this room. Okay, how long it takes you? It takes you really a long time. But if I use that same torch and move it around specific points in the room, it takes probably less time, much less. So this is what Smith-Waterman algorithm does. It takes a pack of, I would say, genetic material, and it will scan it, it will try to create an alignment. So what we do is we create a FASTA file. FASTA is just the format, it's just a text file. FASTA is the accelerated algorithm of Smith and Waterman. And this will help us to understand the content of what has been sequenced using the Sanger device, or the Sanger method. Sanger was a British researcher. In the 50s, he created the genetic sequencing concept, and he used a series of steps. It wasn't an algorithm, really. It was a physical progressing, physical steps, doing this, doing that. It's like a recipe. However, that was exactly taking too long, especially if we had long reads. So the human genome is about 3.2 billion different entries. So if you want to read all of it and try to sequence it, that means identify where is A, where is G, where is T. These are the basic, I would say, components of our DNA. I'm sure you have heard about cytosine, guanine, thymine, etc. So if you identify any anomaly in the output, you could say that this is a mutation. And this is what the FASTA method will help you. Actually, the FASTA is the file structure, but of course it is based on a modified algorithm created by Smith and Waterman. FASTQ is also another format, but it contains the raw nucleotides, which would actually tell me with confidence whether my deciphering is correct or not. So what we are talking about in here, and still why this matters. Matters because if I can understand the morphology of the protein, I can subscribe or prescribe a medicine, I can prescribe a method to resolve that problem. One of the methods is gene editing. Another method is just potentially insertion or intervention at the protein level or at the amino acid level. So this is the machinery that uses ultra-rapid detection of your genome. And here, look, in fast sequencing, normally now it takes 2 to 16 weeks. So if you subscribe to Ancestry.com, they send you the pack, you swab or you put saliva sample, they go analyze it after about 16 weeks or probably 12 weeks, they send you your ancestry. Okay, and this is based on DNA analysis. So, however, in the United States, some of the universities have achieved only 5 hours. And imagine, they started with 11 years, 13 years it took for the first genome to be deciphered. And here, it's taking 5 hours. Why? Because they use high-performance computers, because they use optimized algorithms. And this is where it comes. Computational biology, this is where it comes bioinformatics. Always think about bioinformatics as the database, which I need to use to derive from it relationships, and the computational biology is the way to model things based on that database in order to understand more the relationships. So that's basically how we understand the sequencing of the DNA using artificial intelligence. And artificial intelligence, mainly, we use deep learning to identify the relationships. And when we identify the relationships, in other words, we can create a cluster environment, and we can, in reality, understand predictions. Instead of me reading the next long read, I can really just predict what the next long read will be. And this is why I can optimize. This is why I can reduce the time from years to just hours. And basically, with some of the, if you put a file and you press the button, you press enter, for example, to process that file, in some situations, it's now taking about 5 to 10 seconds. But it really depends. You need to be using a powerful machine. So now we come to the present and the future. We come to DeepMind, and we come to Meta. So DeepMind is part of Google AI. So this is basically the Google AI arm. It was actually established in London. DeepMind is a UK company. They have actually claimed that they are able, or they have already predicted in 2022, 220 million structures of all possible proteins. This is not easy. That means if I put those predicted proteins and I try to compare to reference data, I can create a solution for each misbehaving protein. That means I can create a medicine for each misbehaving case. That's a big claim. So Alan Turing was trying to do that in the 50s, but he couldn't because he didn't have enough processing power. They used to have what? ENIAC in the 50s. It was based on bulbs, you know, computer based on bulbs. Have you seen one of those? It used to occupy a big data center larger than this hotel, just to do processing probably that your mobile is 6 million times faster. So also DeepMind, they tried to predict all the folds, and this is very important in bioinformatics and in computation biology. The fold of a protein, how it is folding, You remember those ribbons? How they fold is very important because this is where you see the anomalies. And if you can predict it, as I mentioned, you can predict also how you can resolve that problem by creating new molecules to treat those problems. So Demis Hassabis is of Greek origin. He is actually the mastermind of DeepMind, and he actually got the Nobel Prize for Chemistry this year just because of his invention of the DeepMind AlphaFold. So the AlphaFold is a system. It's an algorithm, a series of algorithms, plus a computer system using high-performance computing and deep learning and machine learning. So it is able to detect all those and predict all those faults. And also it has the possibility to suggest where the anomalies are and how we can treat that. So now we have another competition, which is from Meta. Meta is the parent company of Facebook and WhatsApp, Instagram, you name it. So they claim that they have predicted 772 million protein molecules. And this is because they have massive power. They have huge back-end systems. They have high-performance computing systems. They have also all those research engineers who are able to create new algorithms or to try and test new algorithms. So this is now competition. So this guy will say, I have 772 million. The other guy, I have 220 or 23 million, whatever. And it is just basically hype. Let's go back to some more reason. Although what they have said is correct, but there is here a very important factor, which is, what is the accuracy of those predictions? According to the standard techniques, the AlphaFold 2, which is the version 2 of the algorithm and the environment, can achieve 90% accuracy. However, the AlphaFold 3 cannot achieve that. At best, it reaches 80%. But in most of the situations, it's about 60–40% accuracy. Besides, they say that using a specific methodology, and forget about the word CASP, think of it as a community, as a blog for methodologies. This is what CASP is in reality. So they say that they have achieved 0.96 angstrom, and forgive me for that, this is a jargon, but this is the distance, and this is the median error. So let's not delve into that. So this means that they have achieved very high accuracy. But how can we verify that? At the moment, we do not have the potential to verify those claims, especially that we do not have what they have in terms of processing power. And then we go a little bit, finally, as a conclusion. Where do we go from there? Or where do we progress? It is clearly that bioinformatics and computational intelligence are setting new standards in our lives. It is true that we are having gene editing. We have even witnessed in the past in hot climate countries, they modified the genes of certain trees in order to sustain heat. There are some studies at the moment which I really am against, but there are certain studies also to modify the genes of certain birds in order for them to sustain heat and sustain extreme temperatures. So gene modification or gene editing is really an ethical problem and an ethical challenge these days. But people want solutions. People want solutions to mysteries, they want solutions to problems, they want solutions to disease, to aging, etc. So what we do, there is always what we call simple common sense. If the risk of editing a gene is less than the risk of a person going through that problem, we should not do gene editing, for example. The other thing is, without the technology, without the power of computing, without GPUs, without machine learning and artificial intelligence, and there is a huge difference. I mean, nobody in 2010 was talking about machine learning and deep learning in the industry. Back then, we were talking about big data analytics, and this stayed until probably 2016. Everybody wanted big data analytics. Everybody wanted Facebook likes. Everybody wanted to change the understanding of, I would say, political realities in order to support this president or that president or prime minister. So the idea is people are moving into trends, and those trends are centered around big ideas. And those big ideas, one of them is bioinformatics and computational biology. Now, and up to probably 2040, it's going to be the era of bioinformatics and computational biology, and even beyond. Because imagine how much Meta is creating and is investing, how much Google, DeepMind, they are both investing. But finally, it is good to resolve problems. It's good to address aging issues. It's good to create treatments, create new medicines. That's absolutely fine. But there is always the ethical issue. How this can be regulated. Should strong regulation be advised, or light regulation be advised? Should people accept that all their data information or all their personal information be shared for research? And how people can abuse that data? We have seen the breaches. We have seen how many breaches we are having every day. We have seen also systems collapsing due to cyber espionage or even data breaches. So it is always a responsibility to work with in these fields. And the responsibility is to strike a good balance between respecting ethical components and ethical, I would say, requirements, and do not also stop research, do not hinder research. So it is not good to have strong regulation, and it's not good to not have any regulation at all. So it is a challenging, really, question. I hope I have presented a glimpse about computational biology and bioinformatics without delving into too many details. I hope you did not hate proteins. Now you're gonna dream about proteins and how the probably amino acids are contending in our body and how you produce proteins. But it is a very, very exciting field of study. And I would welcome now your questions, if there are any. Thank you.
Well, I have a question.
Please.
You were saying that you were against modifying birds, for example, to sustain heat. And I was thinking, if we do this kind of things and these kind of birds now, they can endure heat, they're going to ruin something else in nature, because now that they can stay alive for more than they were determined to, now they're going to eat more. And what they're going to eat. It was not supposed to be eaten by those. So aren't we interfering somewhere where we shouldn't? Because the world is perfect the way God created it, and we are going towards a godless, even society, even with interfering so much into the genes and everything else. We are trying to, even though we are saying that we want to keep balance, I think that if we actually interfere and we actually do the modifications, the balance now is ruined. So what do you think about that?
Well, that's a very, very interesting question. And the interesting about this question is that it touches on ethics, touches on religion, touches on lots of areas. My immediate response would be, let things live freely, without interference, absolutely. But who's gonna agree with me?
Okay.
The idea is when you have big investment by big companies, and by the way, all of this is backed by big pharmas. So all of this research, Meta, DeepMind, they have huge investors, billions of dollars, billions of pounds put by big pharmas. And now all the big pharmas are not designing medicines without passing by the alpha form, without passing by ESM from Meta. So they have them as a system. So it's now becoming like a prerequisite. If you want to design a molecule for a medicine, let's say you want to design paracetamol, okay? Paracetamol, of course, is a molecule. So if you want to design that molecule, you have to pass by Meta or by alpha form in order to have like a green light. So what is that? It's not easy to answer your question. But my personal opinion is leave things as naturally as possible. More questions?
I think one of the problems has been is if you look at the seed industry in the United States, basically, the way that seeds have been manipulated, basically, where they produce a crop, basically once only, you can't reuse any of the seeds from the items that have been grown, basically, and it also comes to the situation where some of the pharma companies have been, how could you say it, using outrageous behavior to stop people using ordinary seeds, basically because they say, well, basically you can't do that because it's based on a hybrid of ours and we own the intellectual property right for that. But I think what people are actually concerned about, that if you were to take the manipulation on the seed industry and apply it to the human environment from that point of view, it would just get out of control. So it's like you say, when you talked about the future with computers, you'll basically have a man and a dog. Basically, the man's there to feed the dog, and the dog's there to keep the man away from the machine from that point of view. So what structures, what boundaries do we put in basically to keep it right from that point of view, you know?
Yeah, absolutely. Absolutely. It's really not, I mean, without the ethical factor, we can have no limits. We reach nowhere, to be honest, because it's going to be a feed-forward cycle, and it's like the viper eating its tail. So what really is happening now is big companies are trying to bring huge, humongous investments in order to create this. They sell the idea. I'm not saying that this is wrong. I'm not saying that this is not happening or it's incorrect. All what I'm saying, we need ethics to manage it. And also, when you see that there are huge factories who are producing GPUs, producing more computing equipment in order to satisfy these. I mean, at one point in time, people are going to be orientated. Now, take for example, if you use Google, they are trying Gemini or Gemini.
Yes.
They tell, yeah, this is an AI tool. If you do a simple search, they try to grab things, and sometimes it disconnects.
Yes.
What's it doing? It's recording whatever you have done. It's sending it and recording it and analyzing it. So, sorry, more questions?
There's one online, George. So if I call from here, you repeat. And
Sure.
Then we can. So, question from Jack. Could you tell us about a recent breakthrough or discovery you have made?
I personally?
That's what it is on the chat.
Okay, so
Please repeat first.
Okay, so the question is, can you tell us about a discovery that you have made? My field is computational intelligence and machine and deep learning and basically in computers. I have backgrounds in genomics, but at the moment I'm working on computational intelligence. So at the moment I am training a large language model for airspace and aviation on a ledger system, if you're familiar with Bitcoin and similar. So this is what I'm doing at the moment. Apart, and of course, among others.
And our NDA prevents you from telling us.
Yeah, I cannot really, I'm under NDA, I cannot really disclose more.
Yes. Humble online, given the black box nature of complex deep learning models, how well could these models explain complex biological processes? Can they really explain?
Okay, brilliant for that question. So the black box model of deep and machine learning or artificial intelligence, how can they explain? So in reality, science is not explainable. And probably I have mentioned that in a previous meeting or session. Science describes, but does not explain. Science tells you that if you heat the water for a certain time at 15 degrees, you increase the heat in the water, it reaches 100 degrees because molecules of gas inside the water are bombarding each other, and then you have water vapor. That's the boiling point. That's what science tells me. But science does not tell me if I wanted to drink a cup of coffee or cup of tea. There is no explainability in science whatsoever. We need another factor, probably from a different dimension. I myself call it spirit, but probably others would call it differently. But yeah, we need to achieve explainability. It's not science. Science tells you how things work, but not how things are used for.
Okay, thank you. There's another question online. Barney is asking, can the slides be shared as a PDF copy?
Yeah, by all means.
So
Can the slides be shared? Yes, of course.
The recording of meeting.
Yeah, why not?
So for those who are new to the meeting, the BCS meetings, the meeting is being recorded and it will be made available by the BCS on the BCS website after the editing. And a copy of the PDF slide will be associated with that as it goes live.
You had a question, sir.
Yes. You know, we saw recently what happened in the American elections, whereby the traditional television houses found themselves in a state of less relevance, and those who leveraged other sources got greater relevance. As a result, a lot of the predictions turned out to be incorrect, but this is the point I'm going to. With the emergence of AI and its evolution, the challenges of ethics, like you mentioned, transparency, data privacy, among others, creates a problem whereby there is now lack of trust. Because it was the lack of trust that is making those guys today, some of them are not contemplating being sold because they no longer have the relevance. Where an individual now on YouTube can have a following of millions of people watching a simple program. Now, what is being done to address this aspect of it? Because I foresee a situation whereby if the ethical side is not sufficiently mobilized along with this thing we're seeing now, we know the guys with the deep pockets, the global organizations, they have the money to throw into these things. Then the people will come to a point and say, I don't trust it. If I don't trust it, it's going to be one of two things. It's either you force it down my throat or I back off from it. So my question is, what is being actively done in order to embed in its truest form, I know it's going to evolve, the consensus of ethics in this AI revolution?
Okay, so what has been done to embed, in other words, verification in the revolution of AI, etc. So verification is a big issue of research in AI. Of course, everyone who has had the opportunity to run an AI program or deep learning program, they know very well that not always good verification techniques are being used. So there is a huge research now happening around verification. But whether this research would translate into commercial actions, that's a different story. Because there are always possibilities to verify the authenticity of the black box. There are always ways to verify, because in reality, we are dealing with input and output. In between, in the middle, we don't know what happens. But there is always a way to verify the input with the output based on certain, I would say, well-known techniques. But the problem is whether they are implemented. I mean, one of the things that happens now is the rudimentary verification, not digital verification. Take, for example, when BBC shows you a footage from, let's say, Ukraine or other places. They say this has been verified by BBC. Sometimes BBC uses artificial intelligence to verify the footages. But in many situations, they use witnesses, eyewitnesses instead. So whether to use this way or this way, both are verifications. But whether we can implement that to become a standard, you need a big change in the mentality, social mentality. Have I answered your question?
Somewhat.
Yeah. What were you expecting?
Well, you're absolutely right. For example, when you talk about algorithmic bias and the issues of transparency,
Of course.
and data privacy,
Yeah.
they are still big issues that in the course of time, it can't even be resolved today. It's in the course of time that would require that. I mean, imagine you are on your PC working, and you just know that you're filling a simple form, and you realize these guys are just trying to take your personal data. You feel somewhat, no, I'm not going to give this, because I'm giving myself away, and that type of thing. For example, I was telling my wife somewhere over there, we bought a little bullet today, blender. The next I knew, on my social media, all kinds of renders all over the place on me. I said, this is terrible. All types.
Will you buy a second one?
And even the third and the fourth one, maybe. All sorts of brands.
Yeah.
So it's an interesting world we live in right now.
Look, this is about social change.
Yes.
truly the background is purely technical. We are talking about advanced technology here, but this has profound implications on our society and how we accept that social change, how things are. I mean, 10 years back, who was talking about artificial intelligence? Nobody. 10 years back, who was talking about ChatGPT? Nobody. Nobody. 10 years back, what was mainstream was social media. Social media analytics. Billions of dollars and pounds were shed just for social media analytics. And now, with a very tiny tool, you could skew your social media output. You could skew it to whatever side you want. So it's really challenging, so to speak. Other questions, please?
I have more, please.
Yes, please.
So I want to know how sensitive these proteins are. You described that let's say that we know how one protein looks like, the one that folds, and if it folds in a different way, it means you have cancer. In case I have flu, for example, is one of my proteins structure has changed? Are they so sensitive, or is it just for major issues?
No. Okay. So let me go a little bit to the concept of transcription. So here, in the concept of transcription, when we loosen the double helix of the DNA has a powerful process to preserve itself. This was designed, an intelligent designer did that for us. Some people call him God. I do call him God as well. But this is what this is what the intelligent designer did in that protein. He put this kind of copying. So you will always have a backup copy. Now, if you lose your mobile, do you have a backup copy of your data? I don't think so. So when you try to, so this is by default, there is a backup copy inside the machinery. And when the transcription happens, there is like a checksum. We call them in mathematics a check digit. Imagine when you are entering your account number on your bank app. If you put the wrong number, the bank app will tell you that there is something wrong. Why? Because there is a check digit in the process. So in here, these exons are like check digits. And then when you reach the coding stage, you are sure that what has been transcribed is going to be translated. And this is why humanity has been able to preserve that over a long period of time. However, factors could affect that. We have factors like alcohol. If you drink too much alcohol, you are affecting your DNA at the very, very, very lowest level. If you drink, I would say, artificial sweeteners inside your juices. Artificial sweeteners have the feature that they go down the mRNA side and they bind to some of those messengers and they can change the messaging protocol. So aspartame, if you know about the sweetener, artificial sweetener, aspartame is a carcinogenic, for example. Why? Because it goes and it binds directly to all those sequences and it will alter that. However, you are always having and we follow that by something called statistical, I would say, majority. So if the statistical majority of those transcriptions and translations was sound, a person does not develop cancer, for example. But cancer will develop if statistical majority of those transcriptions is unsound, and this is where we see this reflected in the sequencing of the DNA. For example, here I have an anomaly, but that anomaly was not accentuated at this stage. I had to amplify that in order to receive this. And how do we amplify that? By using PCRs. If you remember, when people were doing COVID testing, they used PCRs in order to do augmentation up to 50 times. Normally, we do not do augmentation up to 50 times, and this is why many people were saying, oh, we have COVID. But this is wrong, because the augmentation should not exceed 24 times or 25 times. So the PCR augmentation, especially here in the UK, was up to 50, 52. And this was huge. And this is where you can see when you do the augmentation, this is where you can start seeing the anomalies. So this is according to a reference file, so to speak, or a reference structure. This is anomalous. And this could suggest a mutation. And the moment you have a mutation not in the right place, a person ends up by having cancer. Okay? So there is a process of preserving your DNA. And this was by design, right? More questions?
Very interesting.
Yeah, you said you're against gene modifying.
Yeah.
Do you have any reasoning behind that?
Yeah, because look, many people who were part of experiments, let's say you eat modified GMOs, modified foods, such as corn. If you go on diet on a GMO, a genetically modified product, there is a high possibility for cancer to start happening. So the modification in nature on one side is going to create an imbalance on the other. So this is why I do not encourage and don't like at all modifying any genes. And yet, if somebody wants to do that, it's only the doctors who's going to do based on some, I would say, statistical method to weigh the risks. But as a principle, no. That answered your question.
Yeah, I was going to say, for example, you're saying against animals, but then if we're causing, say, climate change and we're heating the environment and we're sort of tailoring the genes of the birds to sustain these more extreme climates, is that not better for the animals?
Not really, not necessarily. But also, the answer is, I cannot give you a 100% answer for that particular question, because there are lots of variables which can, and really the answer would be, it depends. However, my opinion, and this is what I believe in, is to leave things as natural as possible. More questions?
George,
Yes.
does that mean you're opposed to things like gene therapy for sickle cell disease?
I'm not opposed. My preference would be treat it the common sense way, and if the risk is much higher than the risk of that particular gene editing or gene intervention, then why do it? So there is always a risk balance approach. So if you wanna treat someone from a disease, in reality, all of the treatments at the moment are limited. That means if you treat a person for a certain disease, you're gonna cause other diseases. But the problem is, these diseases may not appear immediately on the spot. And they say, oh, this is normal, you are aging. This is normal because you had a previous history of this. Probably there were complications. But they don't say that this is a problem because of that particular change. Yeah.
But surely standard medication also has the same problem where you get side effects. Basically, if you're on long-term medication, you can find there's your body will react to those tablets and cause other problems.
Definitely.
if you look at the blood pressure medication, they are one of the types of medication that causes many problems. Basically, does affect the blood and things like that there within the human body from that point of view. So I think these problems already exist. I mean, you know a little bit about them. The problem with genetics is we're not always aware of the consequences because we still don't have a full picture of everything at the moment.
Correct, absolutely. And to give you also another proof, or it's probably everybody uses it, or many people uses it. You have two types of medicines, and I'm not here talking as an expert in medicine. I'm talking from experience. You have the statins. Statins are used for cholesterol. There is no proof that statins treat cholesterol. So why people take them? The other thing, when you have ulcers or proton, we call them proton inhibitors in the stomach, which would reduce the acidity, like osomeprazole or something like that. All of these do not treat. They mask the problem. They do not treat the underlying problem. So medicine is a big field. Unfortunately, people are trying to use pharma more than medicine. And I'm not an expert in medicine. I cannot comment on those, but what I'm saying this as part of a personal experience. But nevertheless, we hope at one point in time that biotechnology, including bioinformatics and computational biology, will help us to create more commonsensical medicines, hopefully. Fan is telling me time off, time out. Thank you for joining us, and thank you for listening.